let's get into it. Let me just grab a quick sip. So inquiry question is, how does mutation introduce new alleles into the population? So module 6 is a really nice segue from module 5, where now when we look at mutations, firstly we're going back to our processes of replication, translation, transcription, and talking about mutations affecting those processes. Okay, here we are. So first question we have here is that we're going to look at is, how do mutagens operate, the different types of mutations. We're going to talk about somatic versus germline mutation, and then talk about the effects of mutations on coding versus non-coding DNA, and then look at causes of genetic variation, and finish off by talking about population genetics. So we're going to talk about mutations, gene flow, and genetic drift. So here we are. Inquiry question is, how does mutation introduce new alleles into a population? Remember, a mutation, firstly, is a permanent alteration to the nucleotide sequence of an organism's genome. This is where we are permanently altering someone's, or rather, natural circumstances or their own system leads to the alteration of someone's own nucleotide sequences, which then changes the proteins that they are able to make. What exactly happens? So sorry, let me grab a quick sip. Alrighty. So here we are. We're going to now compare the causes, processes, and effects of different types of mutations. But we're first going to talk about point mutations. So point mutations, in terms of what causes them, firstly, they are caused by external mutagens, DNA replication errors, and DNA repair system malfunction. So point mutations are these mutations in the coding sequence. So they just affect, one or two nucleotides. It just affects the coding sequence here, and it can have these specific effects. It can lead to a silent mutation, which means we don't actually see it having an effect on the amino acid sequence that's produced. It can be missense, nonsense. It can lead to the creation of SNPs, which is a single nucleotide polymorphisms, which is the change in a single nucleotide. It can also lead to the creation of new alleles. So mutations are not always bad. They are important for variation, but they can be bad. They can be potentially beneficial, and they can be potentially negative. And just because a point mutation is small doesn't mean that its effects will be small. So cystic fibrosis is an example of that. So here we have point mutation, an example of what actually happens. This is the mechanism, and this is the effect right there. So essentially what happens is that in point mutations, this is a normal codon sequence, right? So we've got, let me use a highlighter. Okay, so we've got AGC, we've got AAG, and GCU. We've got three specific sequences right there, and it produces these proteins, right? So these specific amino acids, which will then join together, form polypeptide bonds, lead to a polypeptide change, form proteins. So there are two types of point mutations that can happen. There can be substitution, or there can be a frame shift mutation. In substitution mutations, what happens is, as you can tell by the name itself, one of the nucleotides substitutes the other. So instead of the G here, the guanine, there has been a substitution, and there's been a uracil that has been inserted. And this, remember, this is happening during translation. So because of that uracil insert, or the transcription has occurred, translation is happening now, but during transcription there's been that change of uracil. From guanine we've gone to uracil, and because of that the code now changes to the codon changes to the codon changes to a the anticodon, my bad. The anticodon changes to AUC instead of AGC, and that means that our amino acid would also change right there. Frameshift mutation can either be insertion or deletion. So frameshift means that it just shifts one over. So it's causing a downstream shift, basically, in reading the sequences. So how does that happen? They can either be insertion or deletion. In terms of insertion, what happens is that we have the ACG. So here we have inserted a C just before the G there, and that has, instead of changing just one amino acid, it's actually changed the whole sequence here. Same thing happens if a deletion happens. So we've deleted the G here, and that has also changed the whole sequence there. And its effects can be the following. This can be the effects of it, right? It can either be missense, which means that in this case here, the mutation edits the mutation which edits codon sequence to incorporate different amino acids in the protein. So there is a different amino acid that has been added in, and that is so that's an example of missense. This is an example of missense, and this is an example of missense. So a different amino acid has been incorporated in. Nonsense mutations, like the nonsense effect, essentially is when a premature stop codon is introduced. So when you look at this, you will actually look at a table that tells you, there's a table that actually tells you what the combinations of these anticodons, what amino acids that they'll lead to. And if you want to check it out, you can just type up amino acid translation table or something. It should pop up, and you could see, for example, that the codon here, AUAG, that is a stop codon. That is telling us that, okay, the process stops. And what it's done is, as you can see, it has prematurely stopped the process. It has prematurely stopped it, and it has prematurely terminated the polypeptide synthesis. A silent mutation, on the other hand, is when, luckily, yes, there has been a change here, but if you look at the amino acid table, you'll see that there can be a variation of sequences, of anticodon sequences, that lead to the production of one amino acid. So AGU also leads to the amino acid SER, that I don't know the full form of SER, but it leads to the production of this specific amino acid, and so does AGC. So it's like so there are a couple of anticodon combinations that will lead to the same amino acid being formed, and so as a result, luckily, there is no effect on the amino acid sequence. Okay, now that was point mutation. That's when the mutation affects the nucleotide sequences. Point mutations is the first one we looked at. The second thing we're going to look at is chromosomal mutations. This is where the entire chromosome is affected. And there are essentially two main causes for it generally. It generally is a result of errors that occur in meiosis, in the process where the gametes are being formed. So essentially, firstly, there could be an issue with crossing over, and if crossing over occurs incorrectly, that means that chunks of DNA can be screwed up, like they can move places, and that can lead to structural mutations, these mutations right here. Secondly, there can also be issues with sister chromatids being incorrectly separated. So remember, when we've got the sister chromatids that line up near the mitotic spindle, that line up and the mitotic spindle is attached to them, they're separated. If there's an issue with separation, or they may not be separated, or they may just be separated for like one side, that can lead to an uneven number of chromosomes, and that leads to mutation in the number of chromosomes. The effects of this are usually severe, more severe than point mutation, because this is where the whole chromosome is affected, so it can affect a number of genes. So these are the structural mutations that can occur. So when the chromosomes go to cross over, for example, it could mean that there's a deletion of a chunk of the chromosome. Inversion, they can like invert over here, as you can see, translocation. A part of the chromosome here can translocate to the other chromosome, and duplication, it can duplicate as well. So it leads to so that's structural mutations. The second one is mutation in chromosome numbers. So for that, there's either aneuploidy or polyploidy. So aneuploidy is when the overall chromosome number of the offspring is different to the parent organism. So in the case of Down syndrome, the individual has 47 chromosomes instead of 46, because they have 3 chromosomes instead of they have 3 copies instead of 2 for trisomy 21, which is chromosome 21. So they have 3 copies of it instead of 2. In terms of polyploidy, what happens is that when an organism contains more than 2 sets of homologous chromosomes, so instead of having 2 chromosomes, they actually could have, for example, 3 copies of every homologous chromosome instead of the 2. And an example of that is triploidy, the condition. An example of aneuploidy is Down syndrome. Okay,